Welcome to this next video in which we are discussing the visual system. Okay, so we've now discussed phototransduction in the rod cells, and we've seen that when light hits one of the rod cells, what will happen is through the cascade that we've spent a lot of time discussing, you will get hyperpolarization of the electrical potential difference across the cell membrane of that rod cell, and that's the electrical signal. So we have turned the visual stimulus, the photons, into an electrical signal, which is this change in the electrical potential difference across the cell membrane, and we'll see how that electrical signal is going to be carried forward later on. What I first want to do is have a look at how this process is slightly different for the cone photoreceptors as compared to the rod photoreceptors. So I want to tell you about the difference between phototransduction in the rods and phototransduction in the cones. Okay, so let's now turn our attention to phototransduction in the cones. So firstly, let me just remind you of the structure of the outer segment of a cone photoreceptor. So remember, this is where the motivation for the names rods and cones come from. The photoreceptor outer segment of a rod cell looks like a rod, whereas the photoreceptor outer segment of a cone cell looks like a cone, so it will look like this. Okay, and remember you have these invaginations of the membrane to get the large surface area, like so. And you can see that it sort of has a cone shape rather than a rod shape. And in the case of cones, all of the receptors for light will actually be in the membrane in the actual cell membrane rather than in membranes inside the cytoplasm, as it was in the case of the rods. So all the light sensitive proteins will be in this membrane here, and that's why we have those invaginations to get as much surface area available for these light sensitive receptors as possible. Okay, now remember there are three different types of cones. There are the S cones, there are the M cones, and there are the L cones, which used to be called the blue cones, the green cones, and the red cones. And these different types of cone photoreceptors have differing sensitivities to the different wavelengths of light. So they're more likely to undergo an electrical signal, i.e. they're more likely to undergo the process of phototransduction in response to photons of light of the appropriate wavelength for each of them. So the S cones are more likely to be activated by blue light as composed to red light. So what I mean by that is you'd have to shine far more red photons onto an S cone to get the same degree of phototransduction as for a few blue photons. Okay, so that's what it means. It doesn't mean that they won't be activated by photons of different wavelengths. It means that the photon wavelength that they're most likely to be activated by is what their title is. So small wavelength, medium wavelength, and long wavelength. And the reason for this difference is that they have different types of light receptors. So we talked about how in the rod cells the protein that is actually sensitive to light is the rhodopsin receptor. Each of these different types of cone cell will have a different type of receptor for light. And I want to now talk about the names for these receptors for light in the cone cells. So, the receptors for light in cone cells are known as iodopsin receptors. Okay, and there are three different types of iodopsin receptor. There is S-iodopsin, there is M-iodopsin, and there is L-iodopsin. So in the S cone cells you're going to have S-iodopsin receptors, in the M cone cells you're going to have M-iodopsin receptors, and in the L cone cells you're going to have L-iodopsin receptors. So, next thing I need to discuss is what is iodopsin made up of. So remember back to rhodopsin. Rhodopsin was made up of a G protein coupled receptor, which was the opsin G protein coupled receptor, bound to that small molecule which was 11-cis retinal. Similarly, S-iodopsin, M-iodopsin, and L-iodopsin are each made up of a G protein coupled receptor bound to 11-cis retinal. So if I take a little piece of phospholipid by there once again, so let's orient this. So this is the extracellular fluid above here, and this is the cytoplasm below here. Okay, so I'm imagining I've taken a little bit of phospholipid bilayer out from one of these slices here, so you can imagine, if you like, cutting out this little bit of phospholipid bilayer, zooming in on it, and this is what we've got. So this is the extracellular fluid side, and this is the cytoplasmic side. So let's now draw one of these iodopsins. So each of them consists of a G protein coupled receptor, and then it's going to have, just like with rhodopsin, an 11-cis retinal molecule bound in. So the difference between these iodopsins and rhodopsin is the G protein coupled receptor, not the 11-cis retinal molecule. We're going to keep that same molecule and use it here as well. So 11-cis retinal. Okay, so what then is the name for the G protein coupled receptors of these iodopsin receptors? So for S-iodopsin, the name of the G protein coupled receptor is S-opsin. For M-iodopsin, the name of the G protein coupled receptor is M-opsin, and for L-iodopsin, the name of the G protein coupled receptor is L-opsin. So this makes complete sense. This is very, very easy. So we saw for rhodopsin that you need the opsin G protein coupled receptor bound to 11-cis retinal. For any of these iodopsins, the S-iodopsin, the M-iodopsin, and the L-iodopsin, the difference is that you use a different G protein coupled receptor. Of course, it will have a very similar function to the opsin G protein coupled receptor, but fundamentally it is a different protein, and the proteins are beautifully named. So S-opsin is the one that you use to make S-iodopsin, M-opsin is the one you use to make M-iodopsin, and L-opsin is the one that you use to make L-iodopsin. Okay, so just to absolutely nail this point at home, if I wanted to make an S-iodopsin receptor, which would be the type of light receptor that I'd have in S cones, I would make lots of S-opsin proteins and bind to them 11-cis retinal molecules, and then I'd end up with lots of S-iodopsins. I'd insert those into the membranes of my cone, and I'd end up with an S cone. Right, so these different types of iodopsin then, they have different preferences for which photon wavelengths will activate them best. So for S-iodopsin, the wavelength of light that is best absorbed is at around 420 nanometers, which is of course in the blue-violet portion of the visible electromagnetic spectrum. For M-iodopsin, the wavelength of light that is absorbed best, the peak absorbance, is at 530 nanometers, which is in the sort of greeny region of the visible electromagnetic spectrum. L-iodopsin, the peak absorbance is at 560 nanometers, which is not in the red portion of the visible electromagnetic spectrum. That's how the names are better, why the cones are better called S cones, M cones, and L cones, rather than blue cones, green cones, and red cones, because really this is not the red portion of the spectrum. The red portion of the spectrum is up closer to 700. This is sort of at the greeny yellow portion of the spectrum. So, just drawing you some graphs as I did for rhodopsin then. So we'll have this graph again, and on the x-axis we'll put the different wavelengths of light, so 400, 500, 600 and 700 here. And what I now want to do is draw the graphs that show how well each of these different wavelengths of light will be absorbed. So really what the probability of a photon of that wavelength being absorbed is. So for S-iodopsin, the graph will look something like this. It'll peak at 420 like so, and then it will go down as we go higher and higher and higher like this. Okay, I will put on rhodopsin as well, just for comparison. So rhodopsin, remember the peak absorbance was at 500 nanometers, so rhodopsin might look something like this, and I'll color code this in. So we'll have S-iodopsin, because that's in the blue cones, we'll have that in blue here. I won't color in rhodopsin. Now what we'll have is M-iodopsin. So M-iodopsin's peak absorbance is at 530, so its absorbance spectrum might look something like this, so its peak will be at 530, but again, the point that I'm really trying to stress with this graph is the fact that do not think that these light receptors, for instance M-iodopsin, will only absorb photons of 530 nanometer wavelength. That's not true. That's the wavelength of photons that it will absorb with the highest probability. It's most likely to absorb a photon of that wavelength. So if you shine two photons, one of 530 nanometer wavelength and one of 700 nanometer, the one that's far more likely to be absorbed is the one of 530 nanometer wavelength rather than 700 nanometers. That's what I hope the message that I'm getting across with these graphs is. So this is the M-iodopsin absorption spectrum here. So I'll colour that one in green, and then finally let's put on the L-iodopsin absorption spectrum. So it will have its peak at 560 nanometers, and its graph might look something like this. And I will colour that in red begrudgingly because it is the light receptor that is in the red cones, as they used to be called, although we should really call them the L clones. Okay, so those are the different photosensitive receptors then that are in the different types of cone photoreceptors. Right, so we have 3 different types of cone photoreceptors, and they have these different types of light sensitive receptors inside of them. Now, the phototransduction pathway is exactly the same now, apart from a few little tweaks, but the principle is exactly the same, that when light activates those photosensitive receptors, the iodopsin receptors, what will happen is you'll get the photoisomerization of the 11-cis retinal. It will turn into all-trans retinal, and that will trigger a conformational change in the opsin G-protein coupled receptor, whether it's S-opsin, M-opsin, or L-opsin. It will then be able to interact with a heterotrimeric G-protein inside the cytoplasm of the cell. Now here's a minor difference that I'm going to tell you about, but isn't particularly important. So the heterotrimeric G-protein that the iodopsins are going to activate inside cones will be different, slightly different from the heterotrimeric G-protein that rhodopsin would activate inside rod cells. Specifically, we will now call it cone transducin instead of rod transducin. And what's the difference between cone transducin and rod transducin? Well, the difference lies in the exact protein that is used as the alpha subunit of the heterotrimeric G protein. So you remember me telling you when we were discussing rod transducin that the alpha subunit was by the name of alpha T1. Okay, so remember, I'll just get the picture back up. Here is my picture of rod transducin. Here we have an alpha subunit, a beta subunit, and a gamma subunit. And look, here's the alpha subunit, and we called it alpha T1. That is the alpha subunit that is important in rod transducin. So rod transducin is this heterotrimeric G protein made up of an alpha, beta, and gamma subunit. We don't care what the beta and gamma subunit are. The important one is that you have an alpha T1 alpha subunit, and as soon as you have that, we call this heterotrimeric G protein rod transducin, no matter what the beta and gamma subunits are. Cone transducin is going to have a different alpha subunit, but it's going to do the same sort of thing, and it's called alpha T2. So it is fundamentally a different protein. It's going to do exactly the same thing as alpha T1. They're both alpha T subunits, and the T of course stands for transducin, and they both do the same sort of thing. Okay, so in cones, our activated iodopsin receptors are going to activate transducin heterotrimeric G proteins of the cone type, and the difference between rod transducin and cone transducin lies in which alpha subunit you have, and in cone transducin you have the alpha T2 alpha subunit. But the process will be exactly the same. It will activate the cone transducin heterotrimeric G protein, it will catalyze the release of the guanosine diphosphate from the alpha subunit, the GTP will come in, and that will cause the conformational change, and I'll just get this back up. That will cause the conformational change in the alpha T2 now subunit, and it will want to cleave away from the beta-gamma complex, and also everything will cleave away from the activated iodopsin receptor. So the alpha subunit will go off, still attached to the membrane by its lipid moiety, and the beta-gamma complex will go off, still attached to the membrane by its lipid moiety. and these can go off and have targets. We're not interested in what the signalling targets of the beta-gamma complex are, we're interested in what the alpha-T2 subunit that is now active is going to go and do, and it's going to do exactly the same as the alpha-T1 active subunit. It's going to go and activate phosphodiesterase 6 inside the cytoplasm of the cone cells. Phosphodiesterase 6 will still be attached to the underside of the phospholipid bilayer. It'll start breaking down cyclic GMP into GMP, and therefore cyclic GMP will go down inside the cytoplasm of the cone cell, and that will lead to the closing of cyclic nucleotide gated ion channels, and therefore hyperpolarization of the electrical potential difference across the cone cell membrane. So there's the difference in the phototransduction process inside rod cells and cone cells, but as you can overall see, it's extremely similar, and the mechanism by which the phototransduction process will be turned off and how you will regenerate the 11-cis retinal is exactly the same between rod cells and cone cells. Okay, right, so we've now got phototransduction, we've finished phototransduction, so I think I'll turn over onto the other side and we'll go from here. So what have we overall seen now? What's the key message? We have seen that whether we're talking about a rod photoreceptor, whether we're talking about an S cone, an M cone, or an L cone, when light activates that photoreceptor, it results in hyperpolarization of the electrical potential difference across the cell membrane of the photoreceptor. That's where we've got to so far. And what we want to see now is how is that message going to be transmitted further? How are we going to send that to the brain? And the other key thing that I want to tell you about is the trichromatic theory of light. So I want to go back and explain how you can actually infer what the wavelength of the light that is hitting a portion of the retina actually is from the information coming from the cone cells. Okay, so that's my plan at the moment. So, firstly, how is hyperpolarization of the electrical potential difference of the photoreceptor going to be transmitted to the next cell along? What even is the next cell along? Well, hopefully you can remember from our discussion of the retina that the next cell along is going to be bipolar cells. And remember, there's considerable convergence usually. So remember, rod cells, you have huge convergence of rod cells onto bipolar cells. Lots of rod cells will all be going onto the same bipolar cell. And you then have convergence of the bipolar cells onto ganglion cells, whereas you have much less convergence for the cones, particularly at the fovea you don't have any convergence at all. And when you do have convergence, it needs to be of all the same types of cones, and we'll add a little complexity to that in just a moment. Right, but firstly, let's just go through rods. How are rods going to pass the signal onto the bipolar cells that they are synapsing onto? So, bringing back up my picture of the retina, if I can find it. Okay, here is my picture of the retina. So here is our rod cell here, synapsing with this bipolar cell. So at this synapse here, the rod cell releases glutamate. So the neurotransmitter that it's using here is glutamate, the major excitatory neurotransmitter within the brain. Now, when the rod cell membrane becomes hyperpolarized because light has activated the rod photoreceptor, the rod photoreceptor will start releasing less glutamate onto the bipolar cell. So glutamate release is actually going to go down in response to activation. And you might think that that's really bizarre because glutamate is a major excitatory neurotransmitter within the brain. It excites other neurons, and here we are, when light comes, we're going to reduce the amount of glutamate we release. However, these rod bipolar cells, they have a strange receptor for glutamate that is actually inhibitory rather than the normal receptors for glutamate, which are usually excitatory. So, let me draw this on here. So, I'm going to draw that synapse between the rod photoreceptor and the bipolar cell out much larger here. Okay, so we'll colour the different cells in here. So we'll have the rod cell coloured in here in red, and we'll have the bipolar cell coloured in here in orange. Okay, so in response to hyperpolarization of the electrical potential difference across the cell membrane of the rod cell, you will get less release of glutamate. So let me draw this on. So here, these are meant to represent synaptic vesicles full of glutamate. So these will be full of glutamate here. And I'll put in a few little dots here to represent the glutamate molecules. So these molecules, these dots inside the vesicles, those are representing glutamate molecules. And how does exocytosis of the synaptic vesicles containing glutamate usually occur? Well, you require a calcium signal. Hopefully you've seen the way that neurons release neurotransmitter in response to electrical signals. Usually what happens is depolarization arrives here, it opens voltage-gated calcium channels, and I'll just draw this on here. So this is a little ion channel. This is representing a voltage-gated calcium channel, or VGCC for short, and we'll have it in pink here. So usually what happens is depolarization arrives in this terminal here. It opens voltage-gated calcium channels. Calcium will come into the cytoplasm of the terminal, and it will then activate the machinery that will cause the fusion of the vesicle membrane with the cell membrane to exocytose glutamate. Here what's actually going to happen is hyperpolarization is going to come to this terminal now, because the whole cell membrane of the rod cell is going to be affected by the hyperpolarization that's occurring in the outer segment, and that's going to inhibit the voltage-gated calcium channel opening. So voltage-gated calcium channels that were previously open are now going to close. So we're going to be getting less calcium coming into the cytoplasm than previously, so we're going to start releasing less glutamate than we were previously. So the amount of glutamate that's being released onto our bipolar cell, and this, by the way, I'll remind you, is a rod bipolar cell. We'll talk about cone bipolar cells later on. So this is a rod bipolar cell. It's receiving information from rods. Okay, so glutamate that's being released onto our rod bipolar cell is going to go down. Now, this is going to produce activation of the rod bipolar cell, and the reason is that the rod bipolar cell has a very strange receptor for glutamate on its surface. It actually has a G protein coupled receptor for glutamate, as I'm drawing here. So this is a 7 transmembrane receptor again, 1, 2, 3, 4, 5, 6, 7. Okay, and this is a receptor by the name of mGluR6. So what does this stand for? Well, the M stands for metabotropic, and then the GLU stands for glutamate, glutamate, and then the R for receptor, and then it's the metabotropic glutamate receptor 6. There are lots of different metabotropic glutamate receptors. I think it's 8 overall, so there's mGluR1, mGluR2, all the way up to mGluR8. On these rod bipolar cells, you specifically have the metabotropic glutamate receptor 6. Now, metabotropic is another name for G protein coupled receptors. And when we say that a receptor is a metabotropic receptor, it implies that it's a G protein coupled receptor rather than an ionotropic receptor, which would be a ligand-gated ion channel. And the major receptors for glutamate that are in the brain, the ones that will excite postsynaptic neurons when the glutamate activates them, they're going to be ionotropic glutamate receptors rather than metabotropic glutamate receptors. So this is a weird receptor for glutamate because it's a G protein coupled receptor rather than a ligand-gated ion channel. And it's specifically this G protein coupled receptor, the metabotropic glutamate receptor 6. Now this will activate G protein coupled receptor pathways inside this cell, okay, and it will lead to the hyperpolarization of the electrical potential difference across the cell membrane of this cell. So I'm not going to go through the G protein coupled receptor pathway downstream of mGluR6 here, but just like the G protein coupled receptor pathway that we've seen inside the photoreceptors, it's actually going to lead to hyperpolarization of the electrical potential difference across the cell membrane of this bipolar cell. So this leads to hyperpolarization. Now, does hyperpolarization produce activation of that bipolar cell? Well, of course, no. Hyperpolarization usually is going to inactivate neurons. Now, bipolar cells don't actually fire action potentials. Remember I said earlier on that the ganglion cell is the first cell along in this sequence of cells that will actually use action potentials. Bipolar cells, just like the photoreceptors themselves, communicate just with what the electrical potential difference across the cell membrane is doing, but hyperpolarization is an inactivation of the bipolar cell. So let's just put this all together. So normally the rod photoreceptor is releasing lots of glutamate onto the bipolar cell, and that's inactivating the bipolar cell. It's hyperpolarizing the electrical potential difference across the cell membrane of the bipolar cell. Now, when the rod cell itself hyperpolarizes, it'll release less glutamate onto the bipolar cell, and therefore you're going to get a break in that hyperpolarization, and therefore you're going to get activation of the bipolar cell. So this is the way that the signal, therefore, is inverted when we're going from the photoreceptor to the bipolar cell. So a hyperpolarization in the photoreceptor is then going to lead to depolarization, or a relinquishing of the hyperpolarization of the bipolar cell, and therefore activation of that bipolar cell. Now, let me then have a look at how the bipolar cell is then going to affect the ganglion cells. Remember, we've had convergence at the level of the rods going to the bipolar cells. So we get lots of rod cells converging onto a single rod bipolar cell. We're also going to get convergence of the rod bipolar cells onto the ganglion cells, the rod ganglion cells. So going back to this picture, here's the synapse that we're now studying, the synapse between the bipolar cells and the ganglion cells. Now, the neurotransmitter that's used there is again glutamate, but this time the synapse or the receptors on the ganglion cell are going to be the more conventional receptors for glutamate, the excitatory receptors for glutamate. Okay, so let's think about how this is going to work. If the bipolar cell becomes depolarized, okay, and remember that's what's going to happen when the photoreceptor becomes activated by light, okay, it's going to become depolarized. It's going to start releasing more glutamate, because you remember previously it was kept in this hyperpolarized state by the continuous glutamate being released by the rod cells. So it's now going to become depolarized, and it's going to start releasing more glutamate onto the ganglion cell, and that's going to activate the ganglion cell, and the ganglion cell will start firing action potentials. Okay, so you'll start getting action potentials sent up the ganglion cell here. Okay, so let's summarize how this rod system pathway is working then. So, we start off with our rod photoreceptors right in the outer layer of the retina. These, when they are activated by light, are actually going to respond by hyperpolarizing, as we've discussed in detail. When they hyperpolarize, they're going to release less glutamate onto the bipolar cells. That seems odd, but until you realize that those rod bipolar cells have a very strange receptor for glutamate. They have the mGluR6, which actually causes them to hyperpolarize in response to the glutamate. So usually the normal glutamate that the rod cells are releasing is causing the hyperpolarization of the rod bipolar cells, and therefore their inactivation. So that stops them from releasing glutamate onto the ganglion cells, and therefore the ganglion cells don't get activated because glutamate would activate those ganglion cells. They've got a more normal receptor for glutamate on their surface. So when indeed we do get light activating the rod photoreceptors, they're going to stop releasing glutamate onto the bipolar cells. That releases the brake on the bipolar cells. They're now going to start releasing more glutamate onto the ganglion cells, and therefore you're going to get activation of that ganglion cell. Now, remember, you don't have rod photoreceptors in the fovea. They're only in the non-foveal portion of the retina, and there is big convergence of rod photoreceptors. So many rod photoreceptors, the number is actually between 15 to 30. So between 15 and 30 rod photoreceptors will go onto a single bipolar cell, okay, and lots of rod bipolar cells will go onto a single rod ganglion cell. So there is that huge convergence. Now remember, I'll remind you, that means that the rod photoreceptor system is extremely sensitive, which means that we can use it for vision under scotopic conditions when light levels are very, very low. We can't use our cone system, we have to use our rod system. And remember, the reason it's so sensitive is that to get an action potential firing along this ganglion cell, you only need light to hit a great big patch of the retina. It's okay for light to hit a great big patch of the retina because all of those rod cells correspond to this single ganglion cell. So you don't need a particularly high intensity of light. You don't need much light to get this ganglion cell to fire an action potential because of the huge number of rod photoreceptors covering a large portion of the retina that all are feeding into that single ganglion cell. So you don't need a high intensity light, basically. So that's why it's so sensitive. Of course, it's at the cost of visual acuity, because if this ganglion cell fires, you don't know which of the photoreceptors actually was activated by the light. And therefore you're not able to trace exactly where that came from as well. So you end up with a much greater possibility of where that light actually came from. It's possible that it came from a larger portion of the visual field than if you were using cone photoreceptors. You can't track it down as well. So lower visual acuity. Okay, so the other thing to say about the rod photoreceptor system is that it becomes saturated under photopic conditions. So remember, photopic conditions means bright daylight conditions. So if you're in bright daylight conditions, then your rod photoreceptor system becomes completely saturated. And what I mean by that is that under daylight conditions, your rod cells will just be being activated continuously, and these ganglion cells from all of the rod photoreceptors all over the retina, they'll just be firing action potentials at a very, very rapid rate, so you won't actually get much useful information at all. So I'll just put this here. It becomes saturated under photopic conditions. It's too sensitive, basically, and it becomes saturated in daylight conditions. So I'll repeat what will happen. So under broad daylight, there's a high intensity light coming in, and it will be going all over the retina, and you're actually going to end up with these rod ganglion cells firing action potentials at their maximum rate all over the retina. Okay? The reason being that the light is too intense, and you've got photoreceptors, rod photoreceptors corresponding to this ganglion cell being activated all the time, and then leading to the activation of this ganglion cell to fire action potentials. That means that you're not actually going to get any decent visual information at all. Because remember, what's important is being able to tell the difference between light coming from different parts of the visual field. That's how you get useful information about your surroundings. If you're saying that all of the ganglion cells from all of the retina are firing at their maximum rate, they're all firing therefore at the same rate, therefore the visual image to you looks just like a great big white mass, effectively. It looks like a clean sheet of paper. You haven't got anything to distinguish different parts of the visual field. As far as you're concerned, with the rod photoreceptor system, the entire visual field looks identical. Okay, so that makes it utterly useless for drawing a picture of the world, the visual world, under photopic conditions. So my overall message from this is that the rod photoreceptor system is fantastic for scotopic conditions. When you're under very low light conditions, say when you're walking around under dusk conditions, that's when you use the rod photoreceptor system. Under photopic conditions, it's completely saturated, and therefore you're not actually able to gain any useful information about the world other than that it's daylight. You know that there's a lot of light there, but you're not actually capable of building up an image of the world because your ganglion cells are all saturated, so there's no distinguishing features between different parts of the visual field, and therefore it would just look like a plain sheet of paper. That's all you would be able to work out about the visual field, a great big white mass, basically. All you'd know is that there's a lot of light there, but you can't actually say that there's anything different from different parts of the visual field. The cone photoreceptor system is what you have to use to actually get a useful image of the visual field under photopic conditions, because it's much less sensitive and therefore doesn't end up saturating in the same way under these photopic conditions. Okay, so I think we'll have a break here. In the next video we'll talk about the cone photoreceptor system, and we'll then talk about where the ganglion cells are actually going to send their axons, and the optic nerve, optic chiasm, and we'll start the central visual pathways.